Hello everyone. So in this session we are going to start with the topic of epitheliums. So epithelium is a very important topic of histology and many questions in the previous year of examination has been asked from this topic. So in this session we are going to know about the epithelium types of epithelium along with the examples, along with the apical portion of epithelium and its modifications, and also the junctions of the epithelium. So all these things will be covered here. So also the previous year of questions being asked for the NEET-PG INI-CET exam will be also incorporated in this discussion. So let's start with this. So first thing which I want everybody to understand, so before proceeding into the classification, it's also important that we should know what does epithelium means. So what does epithelium mean? So it is a sheet of cells which is covering the outer aspect of a solid structure, or it is covering the inner aspect of a hollow or a tubular structure. So it is made of a sheet of cells. Now, other than that, if I talk about the classification of epithelium, it is broadly classified into following types. So if we have got a surface lining epithelium, the major function which is provided is the protection and that of absorption. Glandular epithelium has got a function of secretion, neuroepithelium has got a function of sensation, and myoepithelium has got a function of contraction. So these are the major function of epithelium which you have to know, and it's very important that you should know the example of each variety. So let's start with this with the general features. So before starting with the general features, I just want everybody to know about the epithelium. If I talk about epithelium, epithelium is sheet of cell. It is a sheet of cells that covers the external surface of any solid structures and internal surface of any hollow or tubular structures, So this is the whole definition, you can say, this is a definition of epithelium. Other than that, so this was exactly, so definition of the epithelium I have already written, and let's focus. So let's focus on the different features of the epithelium. So we have completed the definition which I have already written. If I talk about the epithelium, it can be single layered, or it can be multiple layered. Other than that, if I talk about epithelium cells are attached to each other, and that the cells of the epithelium cells is attached to each other, and that attachment is attached to each other by CAM. What is that? Cell adhesion molecule. So that attachment is called as cell adhesion molecules. Other than that, epithelium rests on basement membrane. So if I talk about the component, see here, if you will see the component of the, so epithelium, as I told you, rest on basement membrane. And if I talk about this basement membrane is made of two layers, that is basal lamina and reticular lamina. So we have got two layers of the basement membrane, basal lamina being the part of the epithelium itself, and that of the reticular lamina being the part of the underlying connective tissue. Underlying epithelium, we have got the connective tissue which is traversed by blood vessels, nerves, and other than that, epithelium is having the origin, so it is arising from the three germ layers. So if I talk about epithelium is developmentally arises from all the three germ layers. What does that mean? That means it is ectodermal in origin. As the epithelium of the skin. It is endodermal in origin when we are talking about the epithelium of GI tract or gastrointestinal respiratory tract, and it is mesodermal in origin if we are talking about epithelium of CVS. Epithelium in relation to cardiovascular system. So, this is showing us that epithelium has got origin from all the three germ layers. Now, let us talk about intercellular or junctional complex. If I talk about epithelium, the individual epithelial cells has got the interconnection that is called as junctional complexes. Epithelial cells are adherent to each other by cell adhesion molecule. So, that connection to each other is called as cell adhesion molecules, and we have to understand the connection. So, if I talk about the epithelial cells connection, it is of following varieties. We have got Jonula occludens, we have got Jonula adherens, we have got macula adherens, and we have also got gap junction. So, it is important that we should know about the details of individually all these, that is intercellular junction and junctional complexes. We have to know the details of all this. Now, so done with this, now I will talk about each variety of this, that is Jonula occludens, that is the tight junction. So, we have to talk about Jonula occludens, that is the tight junction, and if I am talking about occludens, it is having no space or gap in between the epithelial cells. Actually, if you will see, if I slightly enlarge this image, so let me just enlarge, what you can see, this is the Jonula occludens, and if you will see here, what you can see, it is present near the apical part of the cell. It is present near the apical, it is present near the apical part of cells or epithelial cells, and exactly if I talk about, so they are occluded, so no space is there. So, exactly it is obliterating the intercellular spaces which is lying between the cells. So, it obliterates intercellular, it obliterates the intercellular space completely. So, it is obliterating the intercellular space completely, and it prevents the passage of material between it. So that and basically if we see the examples where we can see Jonula occludens or tight junction, we have got these present in the intestine and urinary bladder. Now, let us talk about the next, that is Jonula adherens. So, just I am enlarging this. What you can see, this is Jonula occludens tight junction, which is on the apical aspect of the part of epithelial cell. Just you can see lower to it, we have got Jonula adherens. So, just lower to it, you can see Jonula adherens. Now, if I talk about Jonula adherens, the location is lower to or below the tight junction, below the tight junction or Jonula occludens. And here there is a gap which is present of 20 nanometers. So, exactly please write on that. Jonula adherens. Yeah. So, if I talk about Jonula adherens, it lies below Jonula occludens. So, the location is below Jonula occludens. Okay. And also it is important that a gap is there. If I talk about, there is presence of gap. So, there is presence of gap, a gap is there, and that gap is approximately of 20 nanometers. So, अगर हम Jonula occludens की बात कर रहे थे तो वो टाइट जंक्शन था, उसमें इंडिविजुअल एपिथेलियल सेल के बीच में गैप नहीं प्रेजेंट था. बट इफ आई एम टॉकिंग अबाउट Jonula adherens, there is a presence of gap of approximately 20 nanometers, and microfilaments like actins are embedded in it. It is having microfilaments, that is actin embedded in it. Now, the next is macula adherens. The next is macula adherens, which is made of desmosomes and hemidesmosome. Okay, it is made of desmosomes and hemidesmosome. So, macula adherens, if you will see, let me slightly enlarge it. So, this is macula adherens, which is having desmosome, and half of it is called as shape is hemidesmosome. So, hemidesmosome is more towards the basal aspect, and it is, if you will see, macula adherens, it's scattered over the lateral surface of epithelial cells like a disc, and here the gap between the individual cells of epithelium is approximately 30 nanometers, and which is having transmembrane protein embedded in it. So, write down about macula adherens, that is desmosomes and hemidesmosome. So, it is scattered over the lateral surface of, epithelial cells in the form of disc, okay, and it is having a gap of 30 nanometers, and it has got transmembrane protein embedded in it. So, and if I talk about hemidesmosomes are more towards the basal aspect, and its shape is like half of a desmosome, and it is more towards the basal, close to the basal lamina as shown in the image also. Okay. Now, thereafter, let's talk about the gap junction. So, if we see the gap junction, let me just enlarge this. You can see this is the gap junction. So, agar hum gap junction ki baat karte, we are talking about the gap junction. So, gap junction ki baat karte, to it is also present laterally, as you can see, and it is having interconnection, and it causes the movement of the cells, and it is also having a prominent gap, as you can see. So, all these are very important cell junctions which have to be known to you, and that cell adhesion molecule is connecting all these cells. Okay. So, let's now I would like to talk about the surface modification of epithelial cells. Okay. So, if I talk about surface modification of epithelial cells, on the apical aspect there are certain, so exactly यहाँ पे हम apical aspect की बात करते हैं, तो और apical aspect में हमें 4 modification है. On the apical aspect we have got 4 modifications: glycocalyx, microvilli, stereocilia, and that of cilia. So, we have to one by one know about this. So, let's talk about individual aspects. So, let's talk about the individual. So, we have got all these. So, let's talk about. Okay, so let's talk about individuals. So, this is an image where I could like to enlarge. So, we can see the surface modification of epithelial cells. So, here you can see the, so this is the electron microscopic view, and we can see the minute projections are called as microvilli. Actually, the function of microvilli is increasing the absorption, and it is present in the intestinal epithelium. Stereocilia is, and microvilli is non-motile. Stereocilia is also non-motile, but it is more larger and thicker than the microvilli, and it is present in areas like epididymis, and also it is present in olfactory epithelium. Cilia are motile, and it is very important. There are certain differences between cilia and microvilli. For that, I will show you a differentiating table. Cilia are motile, and it is present in respiratory tube and that of fallopian tube, which is very important for not entering any dust particles or foreign structures, organism, and infection inside the respiratory tract. So, it is moving it out by the movements of cilia, and other than that, it is also present in the fallopian tube, where it is important for the movements of the ova, so that exactly the fertilization can take place. Glycocalyx, if I talk about, it is present in the apical aspect, it is present in the intestine, and the function of glycocalyx is concentrating the ions before absorption. So, let's see this. Now, let's talk about one by one. So, we will just, I want everybody to understand that if I talk about microvilli, the microvilli can be of two types. Either it can be of striated border, and the other will be of brush border. So if I talk about the example of striated border, what does that mean? That means that all the microvilli will have equal projections and they are arranged in a regular pattern. और अगर हम brush border की बात करते हैं तो brush border microvilli has got irregular arrangement and it is present in PCTs and it is also present in the gallbladder lining epithelium. So exactly striated का मतलब हो गया equal distribution, regular pattern of arrangement of microvilli. So I just want you to show the example here. So I just want all of you can see this minute projections which you can see here. So these very minute projections which you are seeing here, this very minute projection which you are seeing here is the microvilli. Okay, so we have seen this microvilli and microvilli examples, both striated and brush border, you have to know. So let's write it down. Okay, I will give you the example of each variety, but before that, I just want you to show the difference. So now you can see this is the image where you can see the stereocilia. You can see stereocilias are longer and thicker, non-motile than the microvilli. Stereocilias are present in the epididymis and it is also present in the case of olfactory epithelium. Now, other than that, you can see glycocalyx. For seeing the glycocalyx, electron microscopic view has been shown to you. So, only the microvilli is also looking longer. It is present on the apical aspect of the microvilli. It is present in the intestinal border, concentrating the ions before absorption. Cilia. If we will see here, this is HNE stain cilia are the projection motile and it is ciliated epithelium is present on the fallopian tube and respiratory tube. So, let's write it. So, before proceeding to the next, I just want everybody to write down the examples where all these structures are present.